I would like to introduce the first speaker of the day, was Derek Bartlett. So Derek is an assistant professor at the Eshelman School of Pharmacy at the University of North Carolina at Chapel Hill. He received his PhD in chemical engineering from Caltech, followed by a postdoc in cancer immunotherapeutics at City of Hope. And he then spent 15 years working within industry, where perhaps the most relevant for this talk is his role at Pfizer, where he led the creation of PKPD model frameworks to support the TPD portfolio. And now his research laboratory at UNC combines such mechanistic modeling and cell-based assays as an animal alternative approach to improve the translatability of preclinical studies. So Derek, welcome. The stage is yours, and feel free to share your slides.
Thank you very much for the introduction, and let's see, hopefully that shows. Thank you very much to all the organizers for the opportunity to participate today. Hold on one second. Get my screen back. All right, there we go. Oops, I'm sorry. All right, do you see that okay?
Looks great.
All right, great. So it's a real privilege and honor to be a part of this series, and obviously with the 100th one today, this has just been a fantastic series over the past few years, and I know it's covered a wide variety of topics in the field. Today, what I want to talk about is specifically PKPD modeling for targeted protein degradation. And so I want to start off with some acknowledgments. My current lab members here at UNC, as well as several of the collaborators and colleagues from over the past few years, who it's really been a privilege to work with them in this space, and a lot of their contributions and thinking have influenced many of the topics and insights that I'll be presenting in the seminar today. I also want to emphasize that I have no financial disclosures or conflicts of interest for any of the compounds or programs to be discussed today. And all of these analyses are my own and really intended today just for educational purposes only. And so my laboratory at UNC focused on developing animal alternative approaches to overcome translational gaps in drug discovery and development. And we do this through two main approaches. We combine PKPD, or quantitative systems pharmacology modeling, with dynamic cell-based assays. And we apply this approach to a variety of therapeutic modalities, including targeted protein degradation. And so today, what I hope to accomplish is these two things. First, I want to provide an overview of PKPD modeling for targeted protein degradation. And then second, I want to illustrate how this combination of PKPD modeling and in vitro assays can help guide translational development of TPD therapeutics. So first, let's step back and just consider, so we're all on the same page of what exactly is PKPD modeling. So PKPD stands for pharmacokinetic, pharmacodynamic modeling, and they really establish this quantitative relationship between drug exposure and pharmacological response. So if we think about each component of that, we have the PK exposure. So you might have a drug that's administered to the body. Let's say you have oral administration, so you have an absorption phase, and then you have the distribution and elimination phase. So you have a transient exposure of that drug. We can capture that mathematically through some simple frameworks like compartmental modeling, or more complex ones that are more mechanistic. And then we combine that exposure with the pharmacodynamic response. So we want to characterize what's the effect of that drug exposure on the biological system. And typical frameworks for doing this, you might see referenced as direct response or indirect response. The direct response, basically, we're saying that that drug exposure modulates directly that response. So as the exposure goes up, response increases or is modulated in that way. You can also have indirect response, where you might consider a biomarker. That response may be a biomarker. You have some input producing that biomarker and some output that control the dynamics. And your drug exposure can influence either, let's say, the input or output of that, and thereby modulate the level of that biomarker. So we can have these plots shown here. You may have the drug concentration as a function of time. You understand how that pharmacological response correlates with the concentration of the drug. And then you can combine these two together in sort of that PKPD framework, where you can understand the pharmacological response as a function of time. So really the system dynamics. And relevant for today, then, we might consider for TPD, consider this response as the percent of the protein of interest relative to its baseline level. So in that case, we may consider the production of the protein, its degradation, and the effect of our protein degrader in the cell is to, say, induce additional degradation on that protein of interest, thereby modulating the level of that protein of interest as a function of time. So we don't just want to look at the PK to the degradation, but we also need to understand at the end of the day, the purpose of degrading that protein of interest is to modulate some downstream pharmacology. So we're really looking to link not only that PK to that proximal biomarker of degradation, but then to that downstream pharmacology. So if we think about this schematic shown on the right, you might have a signaling cascade in a cancer cell that's driving proliferation. The protein of interest, contributes to driving that pathway. And so that protein is undergoing its normal homeostatic regulation within the cell, but we come in with a protein degrader. We're able to induce additional degradation of that protein of interest, reduce its level, and thereby alter the cancer cell proliferation. So how do we put this into a mathematical framework? That's the goal of the PK/PD modeling. So our first step at doing this is we really looked at the mechanisms underlying targeted protein degradation. And we just built a basic mechanistic framework around this because we wanted to understand how PROTAC design parameters affected efficacy. And the way we did this is we first started with the PK profile. So we took the protein degrader. We imagined how it would be administered, say, to a patient or a preclinical animal model. And then that protein degrader has to be transported to the site of action, enter into the cells that we're targeting, where it can then form a binary complex with the protein of interest or the E3 ligase. And then it goes on to form the ternary complex, which is necessary but not sufficient for the degradation. And so then it can lead to the ubiquitination and degradation of that protein of interest. So we took relatively simple mathematical expressions for each of these steps, put that together in a PK/PD framework. And with that, we could then simulate, say, protein of interest degradation as a function of time, the binding interactions, things like cooperativity and ternary complex affinity versus binary complex. And we could think about multi-parameter optimization and how that would affect things like the targeted protein degradation. So from this modeling, we could see that PROTAC efficacy was really multifactorial. There's no single formula for success. So you could have an efficacious PROTAC that may have weak binding affinities, but relative or highly efficient ubiquitination. Or on the other hand, you may have one with strong binding affinities, but relatively less efficient ubiquitination. So this was very informative and helped inform a lot of our understanding of the targeted protein degradation process. But in the course of developing these frameworks, we also realized that many times we wouldn't have all the data we'd need to parameterize a fully mechanistic model like that. And we may need a more pragmatic approach where we just wanted to be able to take typical data sets and use them to support translational PK/PD for our protein degrader discovery programs. And so that's where, if you look at this schematic, it's basically the same framework, but we just remove the explicit representation of all of those binding interactions. So we still have that PK profile. And then for the PD, we just have this streamlined version where it's the production and degradation of that protein of interest inside the cell, the normal homeostatic regulation, combined with an induced degradation rate driven by the protein degrader exposure. And so if we put this in a simple mathematical expression, that's just saying the protein of interest, the change in that as a function of time is a balance between its production and its degradation from either the endogenous degradation or the induced degradation from the protein degrader. And that induced degradation rate is modulated by the concentration of PROTAC that we have inside the cell, or if you think about following PK profile. And importantly, we set up this model so that we could parameterize it using commonly measured values from our typical experimental assays that we conduct on these programs. And so this involved these three parameters. So the protein half-life or turnover rate for the protein of interest, and then the Dmax and DC50 values. And from that, we could fully parameterize this model and be able to simulate protein degradation kinetics and conduct dose optimization, et cetera. And so now what I want to do in the remainder of the seminar is take these two frameworks and illustrate how they can be applied in the context of typical data sets that might be generated in the development of protein degraders. So the first case study I'm going to use from the literature, and this is a compound called MS67. It's a WDR5 PROTAC. And this was published from the Jin laboratory at Mount Sinai, and there were also some nice publications that followed up from the Knapp laboratory at Frankfurt University. And here's the structure of MS67. It's a WD5 degrader based on VHL ligand. And what they did in these publications is they went through sort of the types of data sets we would need to parameterize this model. So I want to illustrate the first thing we do is we start with the mechanistic model. We need to think about the PK, which I'll get to in a second. But then once it gets to that site of action, it has to be transported across the cell and then bind and form the binary complex and ultimately the ternary complex. There are a variety of experimental approaches we can use to examine these parameters or determine those parameters. The ones that are illustrated here, we're using the nanoluciferase-based assays from Promega. So this is the nanobret assays for binary binding to WDR5 or VHL, as well as the ternary complex formation. So we can take that mechanistic model, which has the explicit representation of those binding interactions, and we can fit this data and estimate the parameters that we need to extract from this, which are shown here at the bottom, related to the binary binding affinities for the protein of interest, or the E3 ligase, VHL in this case, an estimate of the cooperativity, as well as the cellular permeability. And so these are approximations. If we have more time points or additional data, that can help inform and build additional confidence in these model parameters. But for now, I'll illustrate it using these parameters that were estimated from the data sets shown here. Then the next step, which is a shared step between both the mechanistic model and the streamlined framework, is we need to look at the WDR5 degradation. So now we want to link the exposure in those cells and those binding interactions to then how that drives degradation of the target and ultimately to cell proliferation effects here as our downstream pharmacological effect. So for the degradation, which is shown on the left column here, this is in MV411, so acute myeloid leukemia cells. The data shown by the circles are from the publication, and those are the same in both of these plots. But then I show the fitting using the mechanistic model or the streamlined model on the top and bottom. For the mechanistic model, it's just that single parameter that we're fitting here. For the streamlined model, it's the DC50 and Dmax, and I do incorporate a Hill coefficient in some cases, depending on what that response curves look like. It's more of an empirical fit in this case, but that allows us to capture the experimental data well with the model. So that gives us the degradation. Then we want to relate that to cell proliferation. A nice equation construct that I've found to be very useful over the years in this space, or anything where you have a response that essentially goes between 0 and 100%. This was adopted from a publication that one of my former colleagues, Shinji Yamazaki, had published a little over a decade ago now. And it's the equation here, which I'll call the Ke50 equation, but essentially uses this effect, the E parameter, and in this case for protein degradation, that corresponds to the percent of the baseline protein of interest, or fraction of the protein of interest in this case. And so I fit those data for the cell proliferation here for both the mechanistic and streamlined model that's driven by this WDR5 degradation here at the corresponding concentrations. So now with the PD parameters established from those in vitro data sets, we need to combine that with the PK parameters to build our PK-PD model. And so I've worked with many talented colleagues over the years who can take even in vitro ADME parameters and PBPK modeling and make projections of what the exposures or the PK profile may look like for a compound, whether that's in preclinical species or in human patients. But in many cases, you may have available preclinical PK data or even human PK data available to support the modeling. In this case, I'm using some of the published PK data for this compound. So this is in a mouse here at 75 mg per kg. They gave a single dose, so we can fit that data for the plasma. They also reported the tumor versus plasma exposures in these mouse xenograft models. And so we can actually incorporate an estimate of the site of action exposure, in this case, the tumor, to drive the pharmacological response. So when that's available, that's nice. Otherwise, we oftentimes will use the plasma as a surrogate without other ones being available. So then we can simulate out what the PK profile may look like at the dosing regimen that they use for their mouse tumor xenograft efficacy studies in the preclinical models. And they use 75 mg per kg BID, so twice per day, given 5 days out of each week. And that's shown there. So then what we do is we take this PK-PD model framework that has the in vivo derived PK parameters with the in vitro derived PD parameters, and we can simulate out what that may have looked like and compare that to the actual experimental data that was observed. So the mechanistic model, again, the projections are shown on the top, streamlined model on the bottom. The far left shows WDR5 degradation that's projected for that dosing regimen for both the mechanistic and streamlined model. And then the experimental data shows the qualitative Western blot on the bottom, giving an idea of the WDR5 degradation relative to vehicle control in this case. And then we also can look at the tumor growth inhibition. So there's the relative growth of the tumors in the mice, either without treatment, shown by the black lines, or with the WDR5 PROTAC treatment, shown by the blue lines. And what you can see is with the model, here I show the middle panel shows a fit with a single parameter representing the percent of the cells that are responding in that tumor to the treatment. If I put that at about 80%, we see a very good correlation with the experimental data. Many times you may be using this in a prospective fashion. So you're doing the modeling, trying to anticipate what dosage you may want to use in the in vivo study, et cetera. There you might do something where instead of specifying a specific value, you might want to understand what a range of parameters where you might have some uncertainty, what impact that would have on the projected efficacy and the dose you might want to use in the study. So this is showing if the KPU, which is sort of the relative tumor exposure to plasma and your site of action exposure, how that would vary, how that might affect the efficacy, or your percent responding cells in the tumor, how that could affect efficacy. But now that we have the PK/PD model framework, some of the nice things about this is it not only can facilitate dose selection and help guide experimental designs, but also helps inform PROTAC optimization. So these are just some plots where you can imagine looking at the impact of different parameter values on things like the WDR5 degradation level, the dose that might be required to maintain a certain level of WDR5 degradation, or even as we illustrated in a publication last year, how you could use sort of this multi-parameter optimization and look at what combination of parameters would be required to achieve certain design criteria. And so you can see from this that both the mechanistic PK/PD framework as well as the streamlined one can support this translational PK/PD where we're going from the in vitro to the in vivo setting. But each of these frameworks really should be fit for purpose for the intended application. So when do you want to use the mechanistic framework? That one's most useful if you're trying to make some predictions about, say, how changes in the expression of the E3 ligase or the protein of interest or binding affinities may impact things like the hook effect, potential variability in response, or things like resistance mechanisms, because it has those mechanisms built in. And the mechanistic modeling is very useful to test some of those hypotheses or understand how they may impact the behavior of your protein degrader. On the other hand, the streamlined PK/PD framework is very useful as sort of just a practical approach for supporting if you want to understand how your data from the in vitro setting, it may help you guide sort of dose selection for in vivo studies, or understand where your current protein degrader may stand with respect to even human dose projections or its feasibility for use in certain settings. And so from that, you can make a lot of those projections using very limited experimental data and many times what's already measured on the programs. So now in this second part of the talk, what I want to do is transition to applying specifically that streamlined PK/PD framework, but do it in the context of some of the exciting new data that's starting to emerge with the clinical stage degraders. So again, I have no association with these programs. I'm just as interested in many of as you are to see how these play out in the clinical setting now that degraders have entered human testing. And I'm going to take some of the data sets that have been disclosed in a variety of the presentations at scientific conferences, et cetera, for two different programs and just illustrate how we can apply this data and analyze it with the PK/PD framework I just described. So the first one is from Nurix Therapeutics. So this is NX-5948. It's a BTK degrader, cereblon-based degrader that's given orally to patients. It's in phase 1/2, I think, testing. Phase 1 testing, and they've gone through dose escalation and have put together a really nice package from the preclinical to some of the initial human data for this. So the first step, as I described before, with the PK/PD model is we look at the in vitro data set for degradation as well as proliferation. So this is using that NX-5948 compound at a range of concentrations, looking at BTK degradation on the left and cell proliferation on the right. And this is a TMDA lymphoma cell line. From this, we can extract our core parameters, the DC50 Dmax estimates here, as well as the Ke50 value. And I do have a parameter that corresponds to sort of modulation of the cell growth or proliferation that's fit from that too. And then these in vitro PD parameters are combined with in vivo PK data in this case. So this is the preclinical data in mice. They had different dose levels they were examining, so we can estimate that. This is plasma exposures. We don't have the corresponding, or I don't have access to the corresponding tumor exposure, let's say site of action. So I'm using the plasma exposure as a surrogate in this case. Then we can simulate out the degradation, compare that to the experimental data, and see pretty good correlation in this case from what they observed in terms of endpoint analysis of the tumors. And then we can simulate out the tumor growth inhibition across these dose levels as well. And again, sometimes the modeling works quite well to fit with the experimental data. Other times you have to learn things and there's some refinement needed. In this case, it ended up being pretty close. This is just pure simulation, so no data fitting shown on the right. Pretty good correlation with the in vivo response in that case. Then the question is, so that's the in vitro to a preclinical setting. How about translating to the human data that's been released? So this was recently presented at a scientific meeting where they showed the cycle 1, day 1 plasma exposures for this compound across 4 different dose levels in the human patients. There's some nonlinearity in this. So what I'm fitting here is just from the cycle 1, day 1 data across those different dose levels, and then saying if that were the PK profile and that's maintained, this is what the degradation would look like using our in vitro-based PD parameters across those dose levels. And so we don't see much of a difference across the doses here, but that's also consistent with what they reported experimentally in these patients across this dose range too. So at least at first glance, it gives us an initial look at how this translation may occur from in vitro all the way to humans for this compound. So let's apply this to the final case study, and this one's with C4 Therapeutics. So they have a PROTAC that's a BRAF V600 degrader, also a cereblon-based degrader given orally to patients. And this one, they also put together a nice data set and presented at scientific meetings. So I'll apply the same sort of approach with some additional nuances that are interesting in the case of this program. So here on the left, they reported the BRAF V600E degradation and phospho-ERK inhibition. And so the degradation shows this strong hook effect, whereas you get to higher concentrations, you start to lose the degradation efficacy. So with this streamlined PK/PD model that I described earlier, the way to capture that hook effect without the mechanism represented for binding is just put a simple inhibition term in there. And this was described in an earlier publication that I referenced too. But by including that, we can now parameterize this and basically empirically fit that hook effect that we can then use for the simulations. But what's intriguing is that the phospho-ERK inhibition doesn't show that hook effect. So even though BRAF is coming up, we see the continued inhibition here. And my guess is that may have something to do with inhibition of BRAF from the degrader acting both as a degrader as well as an inhibitor, if that warhead maintains its inhibitory activity. And so phospho-ERK is a common biomarker for the BRAF inhibitors as well. And so you can see that you get that sustained inhibition as you go to higher concentrations. So we can fit those parameters. And then we use that to drive the effects on cell proliferation. Here it's very nice. They have the kinetic growth data, which does give a little more confidence in the estimates for the parameters we can derive from the in vitro data. And so they have the untreated cells shown in black, and then 2 different dose levels here shown at the bottom, as well as a inhibition-only control. And this data was normalized from what they presented. And so we have sort of limited dose range, but we can still extract the Ke50 value to use for simulations in the meantime. And so one of the interesting things just to point out about this combined degradation and inhibition, if that is at play here for any degrader where that might be relevant, is that when you have the combined degradation and inhibition, you actually drive deeper target pathway suppression. So this may be important for certain targets. This is just illustrating with the parameters I showed for that CFT 1946. You can have the degradation at the different levels, and you get some hook effect at the higher exposure, but you drive that inhibition down much farther. And then if you compare one that just has inhibition alone, same parameters at the corresponding doses, you can see with the degradation on top of it, you get further suppression of, say, that BRAF pathway. And so just for illustration, you can think about, say you had a degrader, degrades a target by 90%, so you're down to 10% remaining. And then on top of that, you have 90% inhibition of whatever's remaining as well. So you really bring down your functional protein of interest down to maybe 1% of its baseline value. If you had just inhibition alone and it's still 90% inhibition, you bring it down to only 10%. So it's just an interesting thing to consider with some of these degraders that have still the active warheads that are repurposed from inhibitors. And then what we do is we combine those PD parameters with the PK data. Again, here they have the mouse data in plasma and tumor across different dose levels. And then they applied this as a single dose in mice bearing A375 tumor cells. These are melanoma cell line. And this is pure simulation on the upper right using those PK parameters and the in vitro derived PD parameters. What you can see is it's very interesting. The solid lines are the BRAF degradation. The dashed lines would be the simulated phospho-ERK inhibition, and the circles are the corresponding data that they reported. And what you can see is the model does a fairly good job of capturing the kinetics and the degree of the BRAF degradation, as well as the phospho-ERK inhibition. And then what we did is we went on and also can simulate then the tumor growth inhibition. So this is with those same 8375 cells, now with the same dose levels, just given BID over the course of the study. And again, with just changing that percent of responding cells in a tumor, the one parameter, but using all the other parameters fixed from the previous things, we can capture those responses very well. And again, if we were doing this without the data there and trying to understand dose levels, we could look at a range across those parameters and understand sort of approximate efficacy we may see across the different dose levels. So then the final step is now to look at some of the human data that's been released for this program. And what's interesting is they showed, so there's cycle 1, day 15 data, so more steady state for the plasma exposure profile across 5 different doses that have tested BID in human patients. So even with the nonlinearity, just fitting this experimental data as it is, and then simulating with the in vitro derived PD parameters, we can extrapolate what the potential BRAF degradation or phosphoric inhibition may look like in these patients. And we get some insight that at least preliminary data, this is IHC data that they described from paired biopsies of patients at 80 mg and up. They showed, in this case, probably it's more qualitative, but about a 2-fold reduction in BRAF levels, which would be consistent with what we expect from the simulations, at least. And it'll be really interesting as more data emerges on this program, as well as Nurix and the other companies taking these compounds forward, how this human PK/PD continues to play out for these degraders. And I think we'll all continue to learn about what works best for translating these compounds from in vitro to preclinical all the way to the clinical setting. So just to summarize then, hopefully I've been able to show how PK/PD modeling can help to understand TPD system dynamics and guide this preclinical to clinical translation. And PK/PD modeling is an important component of our toolbox that we have available for this. And it's important to make these models fit for purpose depending on the application or questions you're trying to answer. I think we're all excited to see continued emerging data from the clinical stage TPD programs, and that'll help us continue to build our confidence in our ability to leverage these PK/PD models and preclinical data sets to guide the application of TPD in humans. With that, thank you all for your attention. I'd be happy to answer any questions now or follow up after the webinar.